Alright, in this video we will talk about the fate of pyruvate. It'll be very, very brief. So, fate of pyruvate. Now, we all understand that pyruvate was the end product of glycolysis. Is that not so? So we just put it here. We have pyruvate. Are you there with me? So pyruvate is the end product of glycolysis. Everybody's aware of that. Now this pyruvate has 4 fates. There are 4 things that can happen to pyruvate. Now in the presence of oxygen, if oxygen is present, pyruvate will form what we call acetyl-CoA. Are you there with me? In the absence of oxygen, that is if oxygen is not present, it is going to form lactate. This is the one you are very much aware of. Is that not so? This is what the red blood cell will do. They will form lactate. Then if that cell is in a fed state, pyruvate can form alanine. Are you there with me? If the cell is in a fed state, let me erase it with I want to draw upwards. Okay, if the cell is in a fed state, pyruvate can form what? Alanine. But if the cell is in a let's say fasting state, this same pyruvate can form oxaloacetate. So this one is in fasting state. Are you there with me? So pyruvate has 4 fates. In the presence of oxygen, it forms acetyl-CoA. In the presence of lack of oxygen, that is when in the lack of oxygen, it forms lactate. In a fed state, it forms alanine, and in a fasting state, it forms what? Oxaloacetate. So in the next video, we'll focus more on the fate of pyruvate that forms acetyl-CoA. That one will now lead us down to the Krebs cycle and the PDH complex. Basically, do you follow? So if you are asked to write on the fate of pyruvate, you also have to tell them that pyruvate has 4 fates. 1, in the presence of oxygen, acetyl-CoA. Absence of oxygen, lactate. In the fed state, it forms alanine, and in the fasting state, it forms oxaloacetate. Now there are enzymes for this, so let me just give you the enzymes so we know we can wrap up this video. The enzymes responsible for, let's say, okay, let me just write now for fate 1, in the presence of oxygen. So I will now put pyruvate, right? Pyruvate can form what? Can form acetyl-CoA, right? What's the name of the enzyme here? This enzyme is called PDH complex, pyruvate dehydrogenase complex. Okay, it's a complex enzyme. This is what we will look at in the next video. All right, now in the absence of oxygen, what will happen? In the absence of oxygen, what will happen to pyruvate? Absence of oxygen, what will happen to pyruvate? This same pyruvate can form lactate. Is that not so? That is when oxygen is not available. Is that not so? So what is the name of the enzyme that is here? The name of the enzyme is called pyruvate. Is it pyruvate? No, it's called lactate dehydrogenase. Yeah, it's called lactate dehydrogenase. So the name of the enzyme is called lactate dehydrogenase. Okay, and basically what we do here is that we are going to reduce the pyruvate. So we have NADH forming NAD plus. Okay, so that's what basically happened in this reaction. Now and you know this one will occur more in the red blood cell, and also now for the third fate, which is in the presence in a fed state. Pyruvate can form alanine. That is called a transamination reaction. Pyruvate can form alanine. I will explain this transamination reaction better in our transamination video that will come up protein metabolism. So this pyruvate can form alanine, and the name of the enzyme here is called alanine transaminase. So we have alanine transaminase. There are more details to this, but for now I'm not going to put it here based on this one is for carbohydrate metabolism. Okay, just remember that it's alanine transaminase in a third state. That's the only time you want to form an amino acid from pyruvate, just to ship in something. Alanine, is it an essential amino acid or non-essential? Of course it's non-essential because we just synthesize it from carbohydrates. Are you there with me? So that's why it is non-essential. All right, as we proceed you will get to know more. So for the fourth phase, we have a fasting state. The fasting state, what will happen to pyruvate? This same pyruvate can give rise to oxaloacetate, and if it's forming oxaloacetate, basically what we are doing is you are adding CO2 to it, because basically oxaloacetate is a 4-carbon structure molecule, so we add a CO2 to it. Are you there with me? All right, so the name of the enzyme here is called pyruvate carboxylase. Pyruvate carboxylase enzyme. Pyruvate carboxylase enzyme. So with the help of this one, we will talk about this one when we are talking about gluconeogenesis. Okay, that's when we'll talk about this whole reaction. Now the next thing now we'll talk about will be the PDH complex, and that will be in our next video.